Prokaryotic cells. Cells fall into one of two broad categories: prokaryotic and eukaryotic. Only the predominantly single-celled organisms of the domains Bacteria and Archaea are classified as prokaryotes. Pro equals before, kary equals nucleus. Cells of animals, plants, fungi, and protists are all eukaryotes. CEU equals true, and are made up of eukaryotic cells. Components of prokaryotic cells. All cells share four common components. 1. A plasma membrane, an outer covering that separates the cell's interior from its surrounding environment. 2. Cytoplasm, consisting of a jelly-like cytosol within the cell in which other cellular components are found. 3. DNA, the genetic material of the cell. And 4. Ribosomes, which synthesize proteins. However, prokaryotes differ from eukaryotic cells in several ways. A prokaryote is a simple, mostly single-celled, unicellular organism that lacks a nucleus, or any other membrane-bound organelle. We will shortly come to see that this is significantly different in eukaryotes. Prokaryotic DNA is found in a central part of the cell, the nucleoid, figure 1. Most prokaryotes have a peptidoglycan cell wall and many have a polysaccharide capsule. Figure 1. The cell wall acts as an extra layer of protection, helps the cell maintain its shape, and prevents dehydration. The capsule enables the cell to attach to surfaces in its environment. Some prokaryotes have flagella, pili, or fimbriae. Flagella are used for locomotion. Pili are used to exchange genetic material during a type of reproduction called conjugation. Fimbriae are used by bacteria to attach to a host cell. Career connection. Microbiologist. The most effective action anyone can take to prevent the spread of contagious illnesses is to wash his or her hands. Why? Because microbes, organisms so tiny that they can only be seen with microscopes, are ubiquitous. They live on doorknobs, money, your hands, and many other surfaces. If someone sneezes into his hand and touches a doorknob, and afterwards you touch that same doorknob, the microbes from the sneezer's mucus are now on your hands. If you touch your hands to your mouth, nose, or eyes, those microbes can enter your body and could make you sick. However, not all microbes, also called microorganisms, cause disease. Most are actually beneficial. You have microbes in your gut that make vitamin K. Other microorganisms are used to ferment beer and wine. Microbiologists are scientists who study microbes. Microbiologists can pursue a number of careers. Not only do they work in the food industry, they are also employed in the veterinary and medical fields. They can work in the pharmaceutical sector, serving key roles in research and development by identifying new sources of antibiotics that could be used to treat bacterial infections. Environmental microbiologists may look for new ways to use specially selected or genetically engineered microbes for the removal of pollutants from soil or groundwater, as well as hazardous elements from contaminated sites. These uses of microbes are called bioremediation technologies. Microbiologists can also work in the field of bioinformatics, providing specialized knowledge and insight for the design, development, and specificity of computer models of, for example, bacterial epidemics. Cell size. At 0.1 to 5.0 micrometers in diameter, prokaryotic cells are significantly smaller than eukaryotic cells, which have diameters ranging from 10 to 100 micrometers. Figure 2. The small size of prokaryotes allows ions and organic molecules that enter them to quickly diffuse to other parts of the cell. Similarly, any wastes produced within a prokaryotic cell can quickly diffuse out. This is not the case in eukaryotic cells, which have developed different structural adaptations to enhance intracellular transport. Small size, in general, is necessary for all cells, whether prokaryotic or eukaryotic. Let's examine why that is so. First, we'll consider the area and volume of a typical cell. Not all cells are spherical in shape, but most tend to approximate a sphere. You may remember from your high school geometry course that the formula for the surface area of a sphere is 4 pi r2, while the formula for its volume is 4 pi r3/3. Thus, as the radius of a cell increases, its surface area increases as the square of its radius, but its volume increases as the cube of its radius, much more rapidly. Therefore, as a cell increases in size, its surface area to volume ratio decreases. This same principle would apply if the cell had the shape of a cube. Figure 3. If the cell grows too large, the plasma membrane will not have sufficient surface area to support the rate of diffusion required for the increased volume. In other words, as a cell grows, it becomes less efficient. One way to become more efficient is to divide. Another way is to develop organelles that perform specific tasks. These adaptations lead to the development of more sophisticated cells called eukaryotic cells. Art connection. Prokaryotic cells are much smaller than eukaryotic cells. What advantages might small cell size confer on a cell? What advantages might large cell size have? Section summary. Prokaryotes are predominantly single-celled organisms of the domains Bacteria and Archaea. All prokaryotes have plasma membranes, cytoplasm, ribosomes, and DNA that is not membrane-bound. Most have peptidoglycan cell walls and many have polysaccharide capsules. Prokaryotic cells range in diameter from 0.1 to 5.0 micrometers. As a cell increases in size, its surface area to volume ratio decreases. If the cell grows too large, the plasma membrane will not have sufficient surface area to support the rate of diffusion required for the increased volume. Thank you for watching BookTube channel. Make sure to smash the like and subscribe buttons to receive notifications about new contents.